Okay. Let me get going here. To wrap up, hopefully, the lectures on inferring phylogenetic trees. Just as a reminder, in the last lecture we talked about character reconstruction and then applied the Parsimony character reconstruction method to testing which phylogenetic tree was the best phylogenetic tree for a set of 4 taxa. And in the next lecture on Monday, we're going to start talking about the actual history and diversity and diversification of life, talking about in particular the really big picture, the overall structure of the tree of life itself, and the major lineages of organisms. Most of what we've been covering has been sort of indirectly related to the reading in Chapter 22. There's material in Chapter 22 that sort of backs up the things we've been talking about. There's also material in there that's independent of what we've been talking about. You really should, I listed as reading Chapters 22 and 25. The chapter you really have to know a lot of the detail in is Chapter 22. Focus on that, do the quizzes and other things to learn on it. And on Monday, we're going to do an in-class quiz. And one of the things we're going to cover in that in-class quiz, and I'll send out an email about this later today with instructions about, as Susan Keene said, a note card you can bring in, is going to be this one section in Chapter 22. So it's basically one page. Chapter 22.2, there's one section on testing the accuracy of phylogenetic methods. We're not going to talk about that in lecture, but you need to read it in the book. And then we're also going to ask you some questions of the beginning of the book itself, Chapter 1.1 and 1.2, which relate directly to the lectures we're going to do on Monday and some of the stuff on Wednesday that relates to the major types of cells of organisms on the planet, the general structure of cells and some of the features of cells, because we're going to be talking about the early evolution of life and the major lineages of organisms. And you should have learned a lot of this in BIS2C. And possibly in other courses, but you really need to understand some of those sort of major features of organisms and some of the other sort of general tree of life stuff that's in the first couple of pages of the book. And then starting pretty much on Wednesday, we're going to get into material that's covered in Chapter 26, and I'll give you some more guidance on exactly which parts of that you should learn and what details. I mean, you should read the chapter, but then in terms of what you need to remember from the chapter, I'll give you some guidance on that next week. And we're going to be posting some study guides as well for the different material that you can see what the major topics are and sort of guidance on how to study for the tests. What we're going to do today is really talk about phylogenetic inference and continue on what we were talking about in the last lecture and inferring a phylogenetic tree for those 4 taxa, and then talking about a few different aspects of character reconstruction and phylogenetic inference. We're going to talk about how you root a tree, so we're going to infer the best unrooted tree, and then we have to root that tree, so we're going to talk about that. We're going to talk about which characters are informative or not informative for phylogenetic inference. A related issue is character conflict, so which characters agree with each other, and you'll see what this means in a little bit. And then also a related issue is some of the assumptions that we've been making when doing this parsimony character state reconstruction and phylogenetic inference that we didn't really talk about. We're going to talk about some of those assumptions, and then hopefully right at the end or at the beginning of the next lecture, we're going to talk about an alternative approach to phylogenetic reconstruction, which is called likelihood reconstruction. So I'm going to review just a little bit of what we did in the last lecture, just to bring everybody back into the mood here. So again, we took this principle of parsimony and applied it to phylogenetic inference in this case, rather than just character state reconstruction. And again, what you do here is you compare multiple possible trees. So if you have a set of taxa, there are many different ways you can draw phylogenetic trees to relate those taxa. We are going to test among those different trees and give them each a score. The score is going to be based upon counting the minimum number of character state changes required for that tree given a set of data that we have in our data matrix. And the best tree, the most parsimonious tree, the one that wins, is the one that requires the fewest changes. So I pointed this out before, but I'm just going to point it out again. In lab, and in a part of chapter 22 in the book, they're starting as their default with rooted trees, and using rooted trees to test which tree is the best tree. And this approach has some benefits. One of the things you do when you do this approach is you build rooted trees, and then you identify an outgroup, and you use information about the outgroup, in particular the character states in the outgroup, to tell you which of the character states in your in-group are ancestral and which are derived. And this can be very useful for many reasons, but it is making a very large assumption. And that assumption is that the character state in the outgroup has not changed. That is, the outgroup somehow has these ancestral character states. This alternative approach that we're doing in lecture is actually what most people do in practice in research these days, sort of replaced the outgroup-based method, which is to use these unrooted trees and not make any real assumptions about what the outgroup is when you infer the trees, or what the character states, the ancestral and derived character states were. And as I mentioned before, a benefit of doing the unrooted approach is that for any set number of taxa, there are fewer unrooted trees to compare and give scores to than there are rooted trees. So it makes the actual amount of calculation that you have to do a little bit less, or the comparisons that you have to do a little bit less. So again, we've covered this already, and I'm going to go through it quickly. The general approach is to draw possible trees. For 4 taxa, we can draw all possible unrooted trees. There are just 3 of them. Once you get to a lot more taxa, you can't draw all the possible trees, so you have to use tricks. We're not going to talk about those tricks. But so we're going to just assume you're going to draw all possible trees, and then you're going to take your data matrix, and for each character in your data matrix, you are going to score it on each tree. And each character on each tree is going to get a score that corresponds to the minimum number of changes required for that character on that tree. And then you're going to do this for every character on every tree, and you're going to sum up across all characters for each tree and pick the tree with the lowest score. Again, that's the most parsimonious tree. So here's the data that we used. 9 characters, DNA sequence data. So 4 possible bases, 4 possible character states for each of these characters, and 4 taxa. Relatively simple situation. Usually when you're doing phylogenetic analysis these days, you would have much more data and many more taxa, but we're just going to do the simple case so you understand the methods. And then you can just extend the methods to any data set after that. It gets more complicated, but the principle is the same. So here are the 3 unrooted trees for those 4 taxa. These are, remember, there are only 3 possible unrooted trees for 4 taxa. These are them for those 4 taxa. So we're going to take character 1 from that data matrix. Here's character 1. And again, I already went through this, so I'm going to do it a little bit quickly. And we're going to map the character onto each of the trees, and then we're going to take each character pattern on each tree, and we're going to ask what is the minimum number of changes required for that character on that tree. So here's character 1 on tree 1. We test all possible ancestral character states for that character on that tree. For this particular character, it only comes in 2 character states in the taxa we're looking at. So we only test 2 possible character states for the ancestral nodes in this tree. And the reason we do this is if you tested the other 2 DNA characters, in this case A or G, you would just require even more changes of character state to get to the data that we observe for these organisms. So there's no reason to even test those. They are guaranteed to be less parsimonious than one of the patterns where the character states you actually observe are in the ancestral character state patterns. So there are 2 character states and 2 ancestral nodes. 2 times 2 makes 4 possible patterns. These are the 4 possible patterns. We then make a count for each of them, how many character state changes we need. This one is the winning character state reconstruction. It requires 1 character state change. Any questions about this? Okay, so we did that for character 1 on tree 1. We now do character 1 on tree 2. There it is. There's the resulting pattern. Again, 1 step is the minimum number of character state changes required for character 1 on tree 2. We now go to character 1 on tree 3. You can sense a pattern, as I mentioned before, when you have 4 taxa and 3 of them have 1 character state and 1 of them has another character state. You can always come up with a pattern that requires just 1 character state change. So the minimum, whenever you see this type of pattern for 4 taxa, where 3 of them have 1 character state and 1 has another, the minimum is always going to be 1. You don't even need to do this reconstruction. The minimum is always going to be 1. So now for character 1, we have solved this first part of the problem. For tree 1, tree 2, and tree 3, each of them requires just a 1 character state change. Now we go to character 2. And you do this for character 2 on tree 1, tree 2, and tree 3. Now character 2 doesn't vary at all between the taxa we're studying. So you can come up with a situation when they don't vary in their character state, requiring no changes. It doesn't matter what the structure of the tree is. If everything has the same character state, you can draw the branches however you want. You can always say the ancestral nodes have the same character state as these organisms and therefore have no character state changes over time. So we get 0 whenever the character states are all equal. Now we go to character 3, and we keep marching our way through the characters for each of the possible trees. So character 3 is slightly different than character 1 and character 2. It has this alternative pattern where 2 taxa have G and 2 taxa have A. I went through the character state testing previously for this, and this is the result that you get for tree 1. There are 2 possible character state reconstruction patterns that require only 2 steps. One shown in red, I haven't drawn the ancestral nodes, but the one shown in red would have the ancestral nodes be both G, and then you'd require a change from G to A along here and G to A along here, or the ancestral nodes could both be A, and then you have a change from A to G and A to G here. Doesn't matter which one of those we observe, the minimum number of changes is 2. The good thing about this character for figuring out which tree is the best is that one of the trees, tree 2, is such that we can come up with a character state reconstruction pattern that only requires 1 change. And that's the pattern where there's G at this node and A at this node. So because of the structure of tree 2, we can now come up with we only need 1 character state change to match the data to this tree. For tree 3, we require 2 character state changes. So now again, we're summing up for each character across all the trees. Any questions about this? So we could just continue to march through the characters and do this semi-painful reconstruction. 0 steps for that one because they're all identical. 1 for each of those trees because it's 3 taxa have 1 character state and 1 taxon has another character state. This has basically the same pattern as character 3. Orangutan and gorilla have 1 character state. Humans and chimps have another character state. So tree 2 again scores 1, and tree 1 and 3 again score 2. This is different now. Instead, orangutan and chimp have one character state. Gorilla and human have a different character state. So for this character, tree 1 wins, has a lower score than tree 2 and tree 3. Go to character 8, character 9, and now we sum up across all the characters for each of the trees, and we get one tree that scores better than the other trees. It's like golf scores here, right? So you want the lowest score here, and this is the one that wins. So for this data matrix, for these 4 taxa, there is one tree that scores better, and we consider that the most parsimonious tree. And that's tree 2. This is the taxa pattern in tree 2. Any questions about this? Okay, so now what we're going to do is talk about some associated issues with phylogenetic inference. This, what I did here, and walked you through, is basically parsimony phylogenetic inference. You can now expand it to more taxa, to more characters, to more character states, and it follows the same rules. It gets a little more complicated, of course, but it follows the same rules. So we're going to do a clicker question here. So if you can get out your clickers. So here's a pattern for one particular character and the character states on this particular tree. So suppose we have, well, they might get it right. You can always change your answer. So you have this particular character pattern. Don't know. So for this particular character pattern, how many different patterns do we have to test for the ancestral character states on the nodes here to figure out which of the trees, which of the character state patterns requires the fewest steps? So just like before, we tested multiple patterns for the ancestral character states on those nodes. How many do we have to test for this particular tree to figure out which is the best? So give you another minute or so. You can talk with your neighbors. You can just guess like some people did. All right, another 15 seconds. All right, hurry up. 5 seconds. -2 seconds. All right. Ah, look at that. A lot of different answers here. So the correct answer is E. So let's look at this. Does anybody who answered E want to explain their answer? Yeah.
You can have like A and A, C and C, G and G, so you've got 3 there. Then you can have A, G, G, A, C, A, A, C, and C, G, G, C. So you have 6 more there.
So is there a mathematical way you can explain this?
Maybe 3 squared.
Yeah, so there are 3 character states, and there are 2 nodes. So for node 1, it can come in A, C, or G. And for node 2, it can come in A, C, or G. So we do 3 times 3, and we get 9 possible character states for the ancestral nodes there. Yeah?
What was the mathematical way of figuring it out?
Just 3 times 3.
Where did you get 73 from? Because there are 3 character states.
It's multiplicative, so it's 3 squared would be the, yeah, so the patterns are here. So it can be A as at the first node, and then you have any of 3 other states for the second node, so AA, AC, AG. C for the first one, CA, CC, CG, GA, GC, GG. So what you do, the principle here is that you test the patterns for the ancestral character states for any character state that you observe in your data matrix, in your taxa for that particular character. Are there questions about this now? Does that make sense? If we had A, C, G, and T, how many possible patterns would there be? 16, right? So there would be 4 here and 4 there. It's 4 squared. 16 possible patterns. When we do analysis like this, for example, even when we're using DNA data, the DNA inside organisms codes in many parts of the genome code for proteins. Sometimes we analyze the protein data instead of the DNA data. Proteins come in 20 different amino acids at each site in the protein. So now you have 20 possible character states for each site, and an enormous range of possibilities, even for 4 taxa. And now imagine 20 taxa across thousands to hundreds of thousands of characters. This is why supercomputers and all sorts of interesting computer science is required for actually doing this analysis in practice. Again, we're going to ask you on tests for things that are very similar to what we did here with 4 taxa and testing for these characters with those 4 taxa. We're not going to ask you to do it for 5 taxa or 10 taxa or 200 taxa or what. Yeah?
If you had more than those 2 nodes, say if you had 3 nodes, would you cube it instead of squaring it?
Yes. So if you had 3 nodes, you allow for every possible character state at each of those nodes. So if you drew this out, you could draw all the possibilities, but mathematically what you do is number of character states raised to the exponent of the number of nodes. And when you have real trees like we work on, it's impossible to test all of those because there's just way more possibilities than there are that you could ever search through. So in fact, when people do this in practice, you use special search algorithms that don't scan through all possibilities. That's just impossible. But in practice, that's the principle behind it, is what we've done here in the class.
Question.
Yeah?
So even though we've determined that's the most parsimonious, how do we know where to put the root? Or is it just
That's what we're getting to right now. So good question. To think about for home, this may come up sometime as a quiz or test question, for example. For this pattern, what is the minimum number of steps required for that particular pattern on that data there? So think about that with that relatively simple tree. All right, so now to get to your question, the next thing we have to do now, we started with an unrooted tree, but when we're looking at phylogeny of organisms, we really care about rooted trees. We want to know which taxa are sister groups relative to each other. We want to be able to figure out where the monophyletic groups are in a particular set of data, and you can't do that until you root the tree. You need the rooting to tell you which things are sister taxa and where the monophyletic groups are. And so what you're going to have to do is figure out a way to root a tree. And the way, in general, we root these unrooted trees is to find an outgroup from the data, the tree, the taxa you have in your tree. So if you have 4 taxa, you try and figure out if one of those taxa could be considered an outgroup relative to the other ones. If you remember from class or from the lab, what an outgroup looks like on a tree is the outgroup lineage branches off in the evolutionary tree prior to the existence of the common ancestor of all the other taxa of the in-group. So I'm starting off with a situation that you should be able to figure out. We're not going to do a clicker question that will count for this, but just imagine this was a question on a test. Which of those 4 taxa, I've gotten rid of one of our taxa and replaced it with another one. Which of those 4 taxa would be a good candidate for an outgroup relative to the others? Mouse, right? Why? Does anybody want to explain why mouse should be? About there, I'm hearing many, yeah.
They branched off like way back.
Right, so the other 3 taxa are primates, mice or rodents. If you knew something about mammalian evolution, if you knew anything about the fossil record, if you knew anything about other data that we had for primates and mice, you would be able to figure out, probably, that the 3 of the taxa share a most recent common ancestor to the exclusion of the other one. Now that only works if you know something about the taxa there. So for a test, if we haven't taught a particular group of organisms, we're not going to assume you even know this, which you should know, but we're not going to assume that. But we might ask you on a test, if we've shown you a phylogeny with a group of organisms, we might show you something like this, and you're going to have to figure out if 3 of them are plants and 1 of them is, a squid or something like that. If we've talked about those organisms, you should be able to figure out and remember the phylogeny in order to root a tree like this. So once we find an outgroup, now we can actually do that exercise I showed you before when we were comparing rooted and unrooted trees and actually root this unrooted tree. So I'm going to do it now with the 4 taxa that we have there, and it turns out that there is external data that tells us that of these 4 taxa, the orangutan is almost certainly the outgroup relative to the other 3, just like mouse was. They're just a little more closely related than mouse was to the other ones, but it's still thought that orangutan is the outgroup. And so what we do is we now take orangutan, we draw a root going into the branch that connects orangutan to the rest of the tree, like we did before with rooting an unrooted tree, and now we root that tree. So here's another clicker question. We're going to do, if I can open this back up. We're going to do a clicker question here. So which of these 3 trees, A, B, and C, is the correct tree if we now root this tree here to the orangutan lineage? And again, you can talk amongst your friends here. One of these 3 trees is our winner. All right, another 15 seconds. Everyone done? All right. All right, very good, everyone. So most people, 95% of you chose A, and that's the correct answer. And what I'm going to do is walk you through this in a couple of different ways to really get you to understand what rooting an unrooted tree looks like and to think about it. So A is the winner here. I'm going to show you 2 different approaches to doing this and to comparing the trees in essence. The first is similar to what I did a couple of lectures ago where we draw a branch leading into here. So this is now our root branch and our root node. and we untangle this tree and draw it as a vertical tree. So here's our root branch. We draw it here. We draw the root node. And now coming off the root node, to one side is a branch leading up to orangutan. I'm going to draw it over here. Now, remember, I could draw this on the other side. The nodes can spin around. It doesn't matter where I draw it. I'm just choosing one side to draw it on here for the bottom tree. Here's the next branch. It's a branch coming off of that root node that leads to another node. And then off of that node comes a branch going to gorilla, and another branch that goes to the shared node here. And then one branch goes to chimp, and one branch goes to human. So I've just untangled basically this tree. You want to line up, you want to make sure that you have branches that correspond to each of the branches in the unrooted tree when you do this. So that tree that I just drew is the same as this tree. Are there any questions about this so far? Remember, you can rotate around each of these nodes. So I could have drawn here gorilla going off to the left, or gorilla going off to the right. It doesn't matter. That ball can spin around. It's not changing the meaning at all, which is on the left and which is on the right. Is there a question about this? Or oh, sorry, I didn't see you. Yeah.
Would it be different if, say, the divisions of human and gorilla were switched on the unrooted tree?
Yeah, of course. So if you switch, this is tree 2. If we used one of those other of the 3 possible trees, if we used tree 1 and rooted that tree, it would look different.
Okay.
Does that make sense?
Yeah.
Okay, so I'm going to just show you one more way to think about this, which is to now make sure you can double check, in essence, by taking your rooted tree and comparing each branch one at a time. So here's the root branch. The root branch, let's throw those away. There's the orangutan branch, and you should always be able to line up branches and nodes in some way that they're not crossing over anything. So here's a branch that goes to orangutan. Throw that away. Throw away the next node. There's a branch. Throw away the next node. There's a branch going to gorilla, and so on. You should always be able to line up every single branch and node between the trees. That makes sense here? That's the sort of the way to double check whether or not you've rooted the tree correctly. And again, you can spin everything around. That's why I emphasized before that you have to be able to do these branch rotations in your head. Okay, so now this goes back to your question before. Now, if I had rooted tree 1 at orangutan, it would look like this. And if I had rooted tree 3 at orangutan, it would look like this. So those are 3 different rooted trees now, different topologies. But the one that we said was the winner was tree 2 here. So now we've gotten a rooted tree, finally, out of this unrooted analysis. Okay, so now I want to switch to talking about a little other aspects of phylogenetic inference and the character data that we've been looking at. Yeah. Oh, are you just yawning and stretching? Sorry. So what I want to talk about first is this issue of what's called parsimony informativeness. And what we want to do is look at whether or not characters, particular characters, so this is a concept applied to different characters in your data set. So columns in our data matrix. And we're going to ask, are those characters useful in determining which of the trees is better, that is, scores better than which other tree? So a character is going to be informative if there are differences in the scores between any of the trees, if any one of them scores better than the other ones. And a character is going to be uninformative if all of the trees score equally. So we're going to just go through the characters and see examples of this. So here's character 2. There was no variation in character 2, right? Everything was the same. All trees got a score of 0. So this character is uninformative. It does not allow us to distinguish among the different trees. Got a score, everybody got the same score. So this is a rule now. You can determine that a character is uninformative if there is no variation in that character between the taxa. So here is another character that turns out to be uninformative, which is character 1. There is variation here, but the variation does not allow us to distinguish among any of the trees, right? Each tree scored 1. They required a change, but they all required equal number of changes. And so another rule that we can come up with is if all taxa, if all of the taxa except 1 have one character state, and one of the taxa has another character state, like, oh, sorry, I'll just go back to this one, like this, then that's also not informative. There's, in essence, every tree pattern is going to require one character state change. So these are two sort of rules for identifying non-informative characters. No variation, and all the taxa are the same except 1. And this is going to be very useful to you, in particular, if we gave you a data matrix. I could give you a data matrix of 100 characters. If only 2 of them were informative, you should be able to scan through that data matrix quickly and cross out the ones that are uninformative, and still tell me which tree is the best tree for that data. Because all you have to do is do the character reconstruction for the characters that are going to be informative. So here, for our data set, we didn't have to go through all the characters, in fact. We could have taken the invariant characters, those 3, and thrown them away. And we could have taken all the characters that have just one taxon has one character state and everybody else has a different but the same as each other character state, and throw those away. So we actually only had to analyze 3 characters to figure out which tree was the best tree here. And again, I could have given you 100, and you should be able to figure this out reasonably quickly, which ones are informative and which ones are not informative. So there's a slightly different way of thinking about this. If we gave you more data, this would be important. So if we look at these characters that were informative, it turns out there's a rule that you can apply to everything that will tell you if a character is going to be informative. The character has to come in at least 2 character states, and each of those character states has to be present in at least 2 taxa. So here we have 2 character states, G and A, and each of them are present in 2 taxa. C and T each are present in 2 taxa, C and A each are present in 2 taxa. And if these were your character states, say across 4 taxa or 5 taxa or 6 taxa here, each of these patterns is informative because there are 2 character states at least for each of them, and each are present in at least 2 taxa. So again, you should study this because I can imagine there might be a question on a test where we give you a data matrix and you have to figure out a tree from it, for example. So again, there's the 2 character states, and it's pretty easy to quickly scan through data and find these, or to block out the uninformative ones, either way you do it. Make sense what we're doing here? So this is informativeness of individual characters. So actually we can modify this protocol that I outlined at the beginning for inferring phylogenies by adding a step. Don't do the phylogenetic analysis until you throw away the uninformative characters. You're just going to waste your time if you don't do that. And that's in general what people do with phylogenetic analysis, as we will see for some of the next lectures. When you're comparing a group of taxa and you want to figure out how they're related to each other, you have to find characters that match this type of pattern in order for them to be useful. Any questions about this? Okay. So, I already talked about this. So, when you do this, it's important to realize that the total score that you get will be different. So before we had a total score for tree 2 of 7, because there were 3 characters that had a score of 1 for each tree. The winning tree is still the same. Just the total numbers have changed here. So the important thing is not to give trees a score. The only important thing is to figure out which tree is better than which other tree. Right, so here's when we block out characters, the winning tree gets 4. When we don't block out characters, the winning tree gets 7. But the difference between the trees is the same. Okay, so now we're going to switch to a very related issue, which is something called character conflict. So if you look at the characters in a phylogenetic analysis that are informative, that is the 3 of those 9 characters that we found out to be informative, you can ask a very simple question. Do they all give the same tree the best score? Or do they disagree with each other as to which tree gets the lowest score? If they disagree with each other, they have what is called conflict. The conflict is in which phylogenetic tree the different characters say is the best tree. So for our data, here we had 3 informative characters. 2 of them said tree 2 was the best tree. 1 of them said tree 1 is the best tree. When we add up the scores, tree 2 wins. But not all of the characters support tree 2. So a comparable example of this is that issue of flying and non-flying organisms that we've talked about a bunch. So if we built an evolutionary tree of vertebrates where our only character was flight or no flight, the birds and the bats would group together in our tree. That particular character suggests that birds and bats are related to each other if we only use that to build our phylogenetic tree. Fortunately, our phylogenetic tree of vertebrates comes from a lot of other data. Bone structure, sequence data from across all sorts of different genes, the fossil record, other types of morphology and physiology. That gives us the general structure of the tree. Hundreds to thousands to millions of characters support that tree. And then flight is in conflict with that tree. If we did a reconstruction with flight again, it would tell us a different tree was the best tree. That's the same thing we have here. Character 7 says tree 1 is the best tree. Character 3 and 6 say tree 2 is the best tree. This is really important to realize because biological data is messy. Not every single character points to the exact same answer in every case. And what you have to do is try and figure out either which characters are most useful or add up a lot of different characters and a lot of bits of information to see if you get a consistent signal across a large set of characters. And that's what, in general, people try and do with phylogenetic analysis, is get a lot of information that points to one pattern, not just, say, one character that points to one pattern. That make sense? So, yeah?
So, in that case, would we just say the character 7 is in conflict with 3 and 6?
Yes, sorry, yes, that's exactly what we would say. Character 7 is in conflict with character 3, and character 7 is in conflict with character 6. And character 3 and 6 are not in conflict with each other. They are in agreement, yeah. Oh, sorry, another stretcher. So what can we do when we have character conflict like this? We can gather more data. We can use alternative methods to analyze the data. There are many things we can do. And one of the things we can do, I'm going to talk about in the next couple of slides, something related to this. So one of the issues here that I haven't talked about, which is really important in relation to character conflict, are some of the assumptions, the hidden assumptions that we have made when doing these phylogenetic trees. So I've just listed a few of them here. I'm going to give you an example of one of these, but I'll just go through a few of them before giving an example. So one of the assumptions we have made is that all characters are treated equally. So we're treating character 7 on same footing as character 3 and 6. What if character 7 was actually not, say DNA data, but the structure of the heart, something that is a really sort of core part of the biology of organisms, and characters 3 and 6 were height and hair color. You might want to count the structure of the heart a little more than height and eye color or hair color or whatever when building phylogenies. When you look at data to analyze to build phylogenetic trees, you want to use things that in general are not prone to homoplasy, not prone to having unrelated organisms become similar to each other in that particular feature. And so we don't, in fact, treat all characters equally. Characters are treated quite differently when we do actual phylogenetic analyses in the lab. Another issue is that sometimes you might want to treat different kinds of changes differently. So if you were looking at the structure of the heart, you might think that changing from a 2-chambered heart to a 3-chambered heart was more difficult, less probable than, say, changing from, did the mic just kick out or no? Changing from a 2-chambered heart to a 4-chambered heart. Maybe doubling the number of chambers is easier than changing from 2 to 3. I don't know, but the types of changes that occur you might want to count differently and not treat them all equally. And I'm going to show you an example of this now. I'm not going to go through sort of all of the assumptions that we're making here, but we actually made quite a few when we, by default, treated every type of change as one step equally and every character equivalently. So I'm going to just go through an example here of why you might want to treat different kinds of changes differently. And this relates to the structure and the nature of DNA and DNA replication. So you might have to dig back into your BIS2A memory here. So the DNA bases, A, C, T, and G, come in 2 different major chemical forms. A and G are what are called purines. C and T are pyrimidines. There's a simple mnemonic that I use to remember this if you know what the underlying actual bases are here. So cytosine and thymidine have a Y in their name, and pyrimidine has a Y in it too. That's how I always end up remembering this. But A and G are both have these double ring structures in their chemistry, and the C and T have a single ring structure. They're different sizes, and they pair with the opposite. So when DNA is in the double helix and when you're replicating DNA, purines pair with pyrimidines, pyrimidines pair with purines. And that keeps the total size of the DNA double helix the same by having one of each in the base pairing. When you have a change in the sequence in DNA, there are 2 different categories of changes that people have classified. When you have an A in a particular region of the DNA change to a G, that is a purine changing to another purine, that is called a transition, a DNA-based transition. When you have any other type of change from purine to purine or pyrimidine to pyrimidine is also called a transition. When that happens, you're not changing the size of that area of DNA in essence. It's a simple chemical change. But you can also have a change where a purine replaces a pyrimidine or a pyrimidine replaces a purine. Those are called transversions. And it turns out that in most organisms that have been looked at, for any particular region of the DNA, transition changes are much more common than transversion changes. Transversion changes are selected against and disfavored and in fact less likely to occur even spontaneously without selection. And in the next lecture, what we'll talk about just briefly before we get into the tree of life is what this means, these different rates of change mean in terms of counting number of steps for phylogenetic analysis.